Our next presenter is Dr. DrGiseeleSholler. She's been a speaker here at our conference before. She's a pediatric oncologist at the University of Vermont Children's Hospital. She became interested in neuroblastoma as a fellow at Brown University and devoted her career to this. She has a translational laboratory at the University of Vermont where they're screening new drug combinations to bring forward to new trials, and they're analyzing patient tumors to identify patient-specific therapies. They now have 5 trials open, are available for children with neuroblastoma, and have recently formed a network of hospitals across the United States, and she's chair of the Neuroblastoma and Medulloblastoma Translational Research Consortium. And I asked her to talk today about the various trials that are open at the University of Vermont. Thank you.
Hi, and thank you, Pat, for inviting me to be here today with all of you and to have a chance to introduce to you the work that we're doing and kind of the direction we're heading in neuroblastoma research. Our overall goal at the University of Vermont and within the NMTRC is to develop a translational research program from preclinical testing, analyzing patient samples, and bring these new therapies into the clinic in a relatively quick fashion. Basically, we are testing novel drug compounds at about 20 drugs per year through in vivo in vitro testing. We're also doing genomic analysis of all our patients' tumors and then testing patients' tumors in the goal of developing personalized drug testing and then bringing these into clinical trials. It's kind of the oversight. And our team, this has been an interesting development. It's really, and I can't emphasize enough the parent involvement in how this has happened. And this is kind of just a group of parents that came to the University of Vermont that have really funded all of our work. The Neuroblastoma Alliance, the Ishangala Foundation, Lily's Friends, Friends of Will, that have come together and have worked with pharmaceutical companies to bring these drugs to children, and then the network of hospitals that have come together. This is the members within our consortium. So the clinical trials I'll be talking to you about today are open across the US and getting so even more. Some of these, the last 2 on our list just joined, one last month, one this month. So we now have 10 hospitals. Myself, Dr. Zage, who's here as well at MD Anderson in Texas, Bill Ferguson in St. Louis, Bill Roberts in San Diego at Children's San Diego, Jackie Krivecka at South Carolina Medical Center, Dr. Eslin in Orlando, at MD Anderson Orlando. Deanna Mitchell is at DeVos Children's Hospital in Michigan. Dr. Kaplan is at Living Children's Hospital in North Carolina. And then just in June, Nahal Parikh joined us from Connecticut Children's Hospital. And just last week, Dr. Malampati from Oregon Health and Science University has joined the consortium. So these are the clinical sites for our work. And but what's really important in our consortium has been our translational scientific core as well. So these are PhDs and MD PhDs doing work in the lab, such as Dr. Andre Bachman from the University of Hawaii, who did a significant amount of the preclinical work for the DFMO drug and helped us to bring that into the clinic, as well as Dr. Webb and Dr. Bond, who are focused on bioinformatics and helping us develop our personalized medicine clinical trial. So the preclinical work that we're doing, as I mentioned, basically the goal is to test 20 different agents in vitro and bringing the ones that show promise in combinations into in vivo testing. And then we're also developing an ongoing repository to test against patient tumors that we're able to grow in the lab as well. The clinical trials from all the clinical testing that we've done, and I'm not going to go into too much of the science because I really want to talk about the trials and what's available for your children at this time. Our first trial is a phase 2 trial using nifurtimox in refractory relapsed neuroblastoma. It's also available for medulloblastoma because nifurtimox does cross the blood-brain barrier. And so it treats disease within the brain. We also have and I'm going to go into each one of these trials, a phase 1 trial with a drug called TPI-287, another phase 1 with a DFMO in combination with etoposide, and then our molecularly guided therapy trial, and this is our personalized medicine work that we're doing. So I'm going to go into a little bit about each trial, why we're studying this drug and kind of what the study design of each trial. So the first one, the phase 2 trial of nifurtimox. Nifurtimox was studied now for quite a few years. When I first came here, we had just opened our phase 1. We've completed the phase 1 and are on to our phase 2 testing now. So this is to really look at efficacy of nifurtimox in relapsed refractory neuroblastoma and medulloblastoma. And it's in combination with cyclophosphamide and topotecan. So why nifurtimox? Nifurtimox increases oxidative stress within tumor cells. I don't know if this works or not. Can I show this? So a normal cell has very little reactive oxygen species and is under very little stress. Cancer cells, on the other hand, have at baseline high levels of reactive oxygen species within them as already. And what nifurtimox does is it gets into the cell and it increases the reactive oxygen species or oxidative stress within the cells, and that leads to cell death. So normal cells are not very sensitive to a slight increase in oxidative stress, whereas cancer cells are very sensitive to this. There's been a lot of recent publications looking at oxidative stress in many cancer cells, just suggesting that furthering oxidative stress can exhaust cellular antioxidant capacities and push levels beyond the threshold that leads to cell death. So we're kind of looking on that. And then the last time I presented here, I showed a lot of the preclinical work, which I'm not going to show today, but essentially we showed that nifurtimox decreases tumor cell viability. It kills neuroblastoma cells. And it does so specifically by interacting with catecholamines. Nifurtimox, that's the VMA and the HVA that we all test in the urine. Those are catecholamines that neuroblastoma cells make. And nifurtimox reacts with that to cause essentially more free radicals, semiquinone free radicals and nitroanine free radicals. Results in oxidative stress, leads to cell death, and we showed this both in vitro and in vivo in mice models as well. And also, another thing that it does that's been interesting is that it suppresses something called AKT phosphorylation, which is one of the mechanisms of tumor resistance that neuroblastoma cell has. So it suppresses this, potentially making the cells more sensitive to chemotherapy. In our phase 1 study, which actually was just finally approved for publication just last week, and that'll be coming out in the Journal of Peds Hemonc, we showed that there patients who received at least 2 cycles, we showed that we had 4 partial responses, 5 stable diseases out of 11 patients. And in patients who were able to receive at least 3 cycles. So out of 10 patients, we had 4 partial responses, 4 stable diseases for a median of 6 to 7 months. So 80% of patients did seem to have benefit with this combination. And these are multiply relapsed neuroblastoma patients. Most patients had had at least 3 relapses prior to receiving these. So the COG phase 2 study looking at cyclophosphamide and topotecan alone in first relapse neuroblastoma patients had a 32% response rate. And we feel that already in multiply relapse, we're doing slightly better than this. But to really, now our phase 2 trial does not have a nifurtimox alone window. It combines nifurtimox with cyclophosphamide and topotecan, and we're expecting to increase, or we're looking to see if we can increase on that 32% response rate. And based on our phase 1 data, we are hopeful that we'll be able to see that. So this is an evaluation. Patients receive nifurtimox, which is a pill that's given 3 times a day, continuously, along with cyclophosphamide and topotecan for 5 days, every 21 days. And we are also giving patients with bone disease, Zometa, as well. So in terms of from the phase 1 trial, just looking at some of our patients, this is a patient who received just nifurtimox alone for 6 weeks. And here is the patient disease, and this is within the CNS. So looking at patients who do have CNS disease in the spinal tumor, and then after 6 weeks of nifurtimox alone, we see a significant improvement in the patient's disease. And this is a patient who had significant bony disease, a mass here in the apex of the lung, a mass in the liver, and bony disease. As you can see, this is MIBG scan. And after 6 weeks of nifurtimox alone, we saw significant improvement, especially in the bony disease. And this patient had been on a significant amount of narcotics as well, coming onto this study and was weaned off her narcotics. And then in medulloblastoma, because we are also treating patients with medulloblastoma, we see, again, here 2 spots within the spine that virtually responded above, and then below as well, there's some disease that improved in medulloblastoma. So that study is ongoing, and it's open at 6 of our centers right now. And it's eligible for first relapse as well as multiply relapsed patients with neuroblastoma. All our 10 centers, we're recommending this for patients in first relapse, since there's no other study focused on that right now. And many patients are going on cyclophosphamide topotecan in first relapse, and we're recommending that we add the nifurtimox to hopefully look for an improvement based on historical control. The second trial that we have open is a phase 1 trial with a drug called TPI-287. Now, our kind of look at phase 1 trials, as you've heard earlier, phase 1 trials have a poor, or are not expected to give a significant response. In general, historically, phase 1 trials that are single agent have around a 10% or so, 10 to 15% response rate, whereas phase 1 or early trials that have multiple agents, combination therapies, in general have a better response rate. So we've taken the approach of looking at how can we test drugs and also give the child the best benefit that we can do in one study. So we're doing single agent up front and then combination therapy within the same trial. Generally, the FDA requires that you do at least one, sometimes 2 cycles of a single agent trial, a single agent drug, and that's looking for the tolerated dose, and then it's possible to combine with another drug. So with TPI-287, we did testing to look at what drugs this might be used in combination with, and found that in combination with temozolomide, we saw a significant benefit. So that's the drug that we chose to combine this with. So what is TPI-287? It's a taxane, essentially. It acts, though, as a microtubule inhibitor, so similar to the same category as vincristine, which I know a lot of your children have been on. What makes TPI-287 different from vincristine is that it's been modified to be taken into cells, but not be pumped out by the multi-drug resistance transporter. So that's one of the ways that tumor cells become resistant to drugs, is there's a multi-drug resistant transporter, though, so the drug gets in and the tumor cell pumps it right out. And this does not get pumped out by that transporter. It also allows binding to mutant tubulin. So this is a tubulin inhibitor. Some drugs, in becoming resistant, will mutate their tubulin so that the drugs can't bind anymore. And this does bind to that. This was a few years ago when I was presenting the nifurtimox phase 1 at ASCO meeting. In a poster, the company who makes TPI-287, Archer Biosciences, came to me and said that they have a drug that, in all their drug screening, neuroblastoma looks to be the most sensitive to this drug. They didn't know anything about neuroblastoma, but they asked if we would do some further testing with it. So this is kind of how we paired up, and we helped them develop a phase 1 trial looking at this is some of the works they had done initially. In multi-drug resistant transporter positive neuroblastoma xenografts, they showed, this is the mice model, and this is a tumor size and days, and as these are actually treated with control, and then some taxanes, paclitaxel and docetaxel, which aren't great in neuroblastoma anyway, but they showed a significant delay in growth in the tumors treated with TPI-287. And just some testing where we looked in combination. This is, let me see. So this is control cells, grow at 100%. Cells treated with either TPI or temozolomide at relatively low doses have some effect, but when you combine the two, especially here, we see significant synergy when the drugs are combined together. And so the study objectives of this are to determine the safe and tolerable doses of TPI-287 as a single agent window up front, and then in combination with Temodar. So we're looking for the safety of the drug and how effective can we get it to be alone and in combination. One of the things we liked about this drug in talking with the company is that it has a very wide therapeutic window. So as you heard earlier, usually the FDA will require pediatric trials to start at 75% or lower percent of the adult maximum tolerated dose. So if your therapeutic dose is 100, if your dose is at 100, they're going to require us to start at 75. So they required us to start at so it was 90 milligrams per meter squared, but they had seen effect even as low as 75 milligrams per meter squared. So the therapeutic window is really big. So we were expecting to be starting at a therapeutic dose, at a dose that we expect to see some effect if there was going to be an effect with this drug. So the study design, we do 2 cycles of TPI 287 by itself, and it's given on days 1, 5, and 15. So once a week, an IV infusion of about an hour, and then a week off. So it's a 4-week cycle. And then again, repeated on days 1, 8, and 15, and then a week off. And then the following cycles, cycles 3 and then through 6 on study, patients during that first week also receive 5 days of Temodar, or temozolomide. And these are the dose levels. We're actually at our 3rd dose level right now. And this study, I'll show you at the end where all the sites are in terms of enrolling. But this is being enrolled at 4 centers now, I think. And we are on our, as I said, on our 3rd dose level. This is, I put this there because this, I didn't have the handouts available for you today, but Pat Sullivan has this PowerPoint, any of you who want it, so you can kind of look at what all the procedures and the cycles and days and when your visits are. If you want to get a copy of this, just Pat can send that to you. Or you can email me and I can send you a copy of the PowerPoint too. The next drug that we're looking at is a drug called DFMO. And this again is a phase 1 trial. We're doing it with DFMO and just 1 cycle by itself, and then in combination with etoposide. And both DFMO and etoposide in this trial are oral. So this is an all oral study. And the rationale for this study, so DFMO is really an old drug, actually, just like nifurtimox is an old drug. It's a drug that we've had around for about 40 years for treating Chagas disease. DFMO is a drug that's been around for about the same amount of time, and it's, the other name for it is eflornithine, and it's used for treating African sleeping sickness. What it does is it inhibits an enzyme called ODC. Now, NMYC controls a number of enzymes, and we know how important MYC is in neuroblastoma. One of the enzymes it controls is ODC. ODC, we've shown, and other people have shown, is very upregulated in neuroblastoma cells. And Dr. Andre Bachman in Hawaii did a significant amount of this work and did some really nice papers showing ODC expression in neuroblastoma, specifically in MYC-N, but also in not necessarily MYC-N amplified tumors. ODC leads to the production of polyamines. Polyamines cause cell growth and proliferation. So inhibiting this can inhibit cell growth and proliferation, specifically in neuroblastoma cells, since this is upregulated in neuroblastoma cells. So we collaborated with Dr. Bachman in Hawaii and started to test DFMO and different drug combinations to see what chemotherapy. We obviously didn't want to necessarily do just a single agent trial. We wanted to see what would be the best combination to use. And so etoposide is a drug we know is effective in neuroblastoma. And looking at these combinations, this is, so these are control cells. These are 3 different pretty aggressive neuroblastoma cell lines. With etoposide alone and DFM alone, we see some effect, but when we combine the 2, we saw a nice cell kill happening. And then in our mice models as well, the DFMO and etoposide curve showed a significant slowing of the neuroblastoma tumors on the mice models. And so we've taken this forward into a phase 1. This opened just a little bit earlier this year. We're now on our 2nd cohort of this trial. And again, it's to look at the safety and tolerability of DFMO as a single agent drug and then in combination with etoposide. We're looking at progression-free survival and response rate and measuring pharmacokinetics in this tumor. We're also, the biology studies I haven't mentioned on my other trial. So all our studies include biology. We're isolating neuroblastoma cells from patients' bone marrow every time a patient has a bone marrow on any of these studies. And looking at genomic expression profiling of those tumor cells if we're able to isolate them. We're also, in this study, looking at polyamine depletion, since we know that this enzyme, this drug, blocks ODC and then should therefore lower polyamine levels in patients' urine and in the tumor cells. So we're looking at measuring those as well. So again, it's a multi-center dose escalation study. We're doing 3 patients at each dose level, and right now we're on our 2nd dose level. The oral DFMO is given twice a day, so it's a pill that the child takes twice a day, continuously. There's no breaks. And then that's for 21 days as the first cycle. And then starting in the 2nd cycle, the patient continues with the DFMO twice a day, and then adds in etoposide, and the etoposide is 50 milligrams per meter squared, daily dose for 14 days, and then a week off to recover. And the DFMO is in a powder that packages that are put into, basically put lemonade is what they recommend, or apple juice. And this is the first drug that the children have told me that I said it wasn't going to taste like anything, and it really doesn't taste like anything. So the kids have done really well in drinking the DFMO powders. This is our kind of dose level. We're starting at 500 milligrams per meter squared twice a day, and we're going up to 1,500. In the adult trials, they went up to 30 grams. This is a drug that's also used in adult colon cancer specifically, and in colon cancer prevention. And they've shown that there's a significant wide window of tolerability. We are looking for effect of the drug, not necessarily toxicity. We don't expect to see significant. We're not going to continue dose escalating past this. We expect at these dose levels we should see polyamine depletion and effect of inhibiting the ODC, and that's really what we want to look for. The one concern is that the higher doses can cause hearing. It's reversible hearing issues, and many of our patients have previous hearing issues because of the cisplatin that they've received earlier. So patients do have audiograms done with every cycle to monitor for that. In the adult trials, it was reversible, so we expect that if we hold the medication, things will reverse. But that's something that, although at these dose levels, the adults didn't see it, the adults weren't previously having hearing issues. So then there may be something to watch for. And again, this is, I just put it in there so you would have it as a handout, but this is kind of when we need to see the patients, basically weekly, and kind of weekly physical exams, especially through cycle 1, and then every other week through cycles 2 through 5. But the clinic visits for this one are relatively short because it's all oral. There's no IV infusions. The kids don't need significant amount done. So it's been a nice one for some patients. And so then the final study that I wanted to talk a little bit more about is our feasibility trial using molecularly guided therapy for the treatment of patients with recurrent and refractory neuroblastoma. And this has been a trial kind of in the works for the past couple of years, and the one that we're most excited about. And you know, it's still kind of just taking our first steps, but this has, with the help of the Neuroblastoma Alliance and Lily's Friends Foundation, and actually now the Asian Gala Foundation, has helped to move this forward much faster than we had expected. And so it's really looking at the right drug for the right person at the right time. So can we do personalized medicine? And we're still asking that question. We haven't defined that question yet. But essentially what we're saying is, you know, standard therapy, most people, significant number of patients respond to, but that there are patients who are non-responders. And who are they, and can we analyze them specifically? If we look at, these are just some bone marrows from patients that we've isolated, just showing a little bit, you know, patients are different. Some patients have more mature looking neurons that have long axons. Some patients are more neurosphere-like. Some patients are a little bit of both and have neurospheres that kind of grow off. But these patients are different. They're all neuroblastoma patients. And then how can we, you know, even just from tumor biopsies, when we grow the tumor biopsies in culture, you know, same culture conditions, but they grow differently. So how are these patients different, and can we figure that out? We've shown that we can take neuroblastoma from the bone marrow, put them into mice, isolate the tumor, and the patients grow. We can create mice models of patients. Can we use that for drug testings? So over the past couple of years, we've had over 70 neuroblastoma samples of different patients. And each one of these rows represents a gene chip analysis of a patient. So we're using the U133 RNA expression profiling chip that looks at over 22,000 different genes in a patient. And you know, these guys look similar, but they look very different from this group of patients. And this group of patients looks different. And can we take each of these groups and find out how should we be treating them separately, differently? And Or should we be treating these patients? Right now we're treating those patients the same as we're treating these patients, but their expression profiling is different. Can we pick out this one patient and see what's different about him? How does he cluster out differently, and what pathways are red is upregulated? This is the NF-kappa B pathway that was very upregulated in that patient. And then there are drugs that can target those pathways, such as Velcade would target that pathway. And can we do that for each patient? So that's kind of brought us to where we are now with a feasibility trial. And this is, is it feasible to take a patient's bone marrow or tumor biopsy, and perform a genome-wide expression profile on this patient and make real-time treatment decisions? Because our patients can't wait. We can't wait 5 months. Everything will be different by then. We'll have treated them with something else, and their expression profiling may be completely different. And then can we look at response rate and progression-free survival? And I say progression-free survival ratio because this is something we're doing in collaboration with some of the adults that are a little bit ahead of us, as usual, on this. Dr. Van Hoff at TGen in Phoenix did some Bisgrove trials, adult trials using personalized medicine, and his endpoint was the progression-free survival ratio, because if we're treating every patient with a different chemotherapy, how do we know if this method is effective? And so he looked at what the patient's response rate was on their previous relapse therapy, and then what it was on his predicted relapse therapy. And if it was greater than if his predicted therapy was greater than 1.3 times longer, their progression-free survival, than what was on the standard relapse therapy, he predicted that it was effective. It's a new endpoint for clinical trials, and I think it still needs a lot of looking at, but we're going to be measuring that on all our patients. So our study committee for this trial has, the core bioinformatics. Dr. Webb at the Van Andel Institute has really been the pioneer of a lot of this work. And I'll explain a little bit of what he's done and how he's developed the informatics modeling, so the computer analysis of these tumors, and how are we analyzing this and coming up with drug profiles. And then Dr. Bart Kamen is he's a pediatric oncologist and pharmacologist, and he's head of the Leukemia Lymphoma Society. He will be analyzing our treatment regimens after the tumor board comes up with this for each patient, looking for safety, because these treatment regimens may be drugs that have never been combined before. And we do have 2 pharmacists on our team to help us put those drugs together. And Dr. Javed Khan from the NCI runs their oncogenomics group there, is also doing DNA expression analysis, DNA profiling, next-gen sequencing of all our patient tumors, and has been a really key player in putting this trial together. So this is an open-label, multi-center trial, but it's a feasibility trial. We're not looking at efficacy yet at this point. What we're doing is we're enrolling and consenting patients, and this first, we're saying that we need to do this in less than 2 weeks. We need to consent the patient, do the biopsy, run the RNA expression analysis, generate a report, have a tumor board meeting, and our tumor board is our 10 centers across the US, including the NCI, actually. So you're going to kind of, how do we say this? Consultation from 10 hospitals, including the NCI, to review the patient's case, every therapy, then everything that's happened with the patient up until this point, the tumor board report, and create an individualized treatment plan that we think is the best therapy for this patient. That gets then reviewed by the medical monitor within 24 hours for safety, and then the treatment regimen is presented to the family. So all this needs to happen within 2 weeks for this to be feasible to do this in your clinic. And then the patients will then be consented for that treatment plan. And we want to see, do patients consent to this or not? Is this something that that is kind of received, and are we able to actually treat the patients? And then the patients are treated, and they're seen weekly for safety visits, and reevaluated every 2 months for efficacy. So I'm not going to go too much into this. This is a little bit of how we're doing the analysis. So the patient's tumor, we receive an expression profile that looks at 22,000 different genes that are upregulated, downregulated, what's going on within your tumor. And then how do we compare this? First, we have to compare it to something normal. So what we've done is use normal sympathetic neurons as the control. So this is the tumor cell of origin, normal sympathetic neurons. And how have they changed? How has this now become a cancerous cell from what it was before? So what's changed to make it cancerous, and what pathways have now changed up or down? And that gives us a Z-score. And then there's multiple different methods. I'm not going to go into all of them that in this computer program predicts which drugs. For example, if there's, I mean, just simply, if the target is there, if there's a drug target like the EGF receptor is present on your child's tumor, and there's a drug that targets that, that gets pulled into the predicted methods. If this, and then we do topological enrichment, which looks not only as the receptor present on your child's tumor, but are all the genes downstream? So is that pathway active? And if that pathway is active and driving the cancerous growth, are there small molecule inhibitors and other drugs that can target pathways as well as just receptors? And then there are a few other pathways, the connectivity map, and there's the NCI 60 drug screen. And that's the NCI took 60 tumors, and they tested chemotherapies against them. And then did expression profiling of all those tumors and classified them as either sensitive or resistant. And so there's a fingerprint of what looking at expression profiles, and the patient's expression profile is matched to see where the fingerprint falls. So does it have a fingerprint that predicts that cyclophosphamide will be sensitive, or is it resistant? And that's how the chemotherapies kind of get pulled into this one. There's the targeted therapies, and then there's the chemotherapies. And then we have a report. And if a drug is predicted by multiple different methods, so the connectivity map, which I didn't talk about, is another method that Broad at MIT uses. So this computer program uses all those different methods. And if it's predicted by multiple different methods, it gets pulled much higher. It gets a higher score, gets pulled to the top. And from those, we create a treatment plan. And we go through all of this with the families, too. And so our IRB had asked us to do first a 5-patient pilot for feasibility just to this point. Can we actually do this in 2 weeks before we start presenting treatment plans to patients? Although they did say that if the patient had no other therapeutic options after doing this, we are able, even in this first 5-patient pilot, we were able to present the treatment regimen. The patients were able to go on those therapies. These are all FDA-approved drugs, and so we're able to prescribe them. And so 2 patients from the first 5 are actually following our treatment regimen. And others were on other studies, so it was kind of biopsies were being done, and so we went through. So the first 5-patient pilot has been completed. All 5 patients were able to be done within 2 weeks. Everything went very smoothly. The tumor board was able to come up with appropriate treatment plans for all patients. And now the NCI, Dr. Javed Khan has invited our entire consortium to the NCI on July 19th and 20th in a week or so. And we're going to do a full analysis of all the 5 patients. And then from this, report this back to our IRBs, and then we hope for the actual study to be open to we're actually treating patients. Likely will be end of August by the time that actually happens. And the validation portion of this, so all patients, we're doing biopsies, all the cells are going into culture as well as being RNA expression profiled. So for example, for these first 5 patients, all of the patient tumor cells are being grown in culture right now. And then we are doing drug testing using the drugs that were predicted for this patient on their tumor cells in culture. And then all patients were also injected into mice for in vivo drug testing. So if the tumors all grow in mice, we'll be testing the mice with these drugs as well. And then we're also looking at genomic validation. And this is something that will take me a while to explain. But in brief, if we take the patient's tumor cells and look at the expression profile and then treat them with drugs 24 hours later, we can redo the expression profiles and see if we wanted to suppress this pathway at 6 and 24 hours after drug treatment, redo the expression profile, and was that pathway suppressed? So that's another method of validation that we are going to be doing with each of these first 5 patients. And then we'll be analyzing these to see what would be the best validation method to use in an efficacy trial to see if we can prove therapy. And then are some drugs better predicted than other drugs. We want to compare drug to drug as well. So this is just an example for the first patient that we did, and needle biopsies were done. All of these are needle biopsies. They're not open biopsies. They're ultrasound guided that we've been able to do. So biopsy 1, that went to in vitro drug testing and into a mouse. Biopsy 2 of the same patient went to the NCI for the DNA sequencing with JavaCut, and we also send samples to Yale to CHOP for ALK mutation testing. Since we're doing the biopsy anyway, we should get as much information as we can for the patient. And then RNA collection for gene chip analysis at UVM, and RNA collection for gene chip analysis at our research lab. So for it to be used in a patient, it needs to be done at a CLIA certified research lab. So that's the one that's actually gives us the report. This is kind of our needle biopsy. It's a small biopsy, and this is patient number 1's neurosphere is growing in culture. And so so that study is in between right now. It's still technically open. It's just we're not fully open it's on hold while the analysis is being done, and then we expect that one in September to be open at all our sites, hopefully. This nifurtimox cyclotopo study right now is open at these 6 centers. It's going through the IRBs at our other centers. It's just not fully open at all centers right now. TPI 287 is open at 4 centers right now. That one will be open in North Carolina very, very shortly, probably this month. And then the DFMO etoposide is currently only going to be open right now at UVM in Orlando. And then Children's Hospitals Orange County is not part of our consortium, but they were very much involved in the early development of DFMO, and Dr. Lenny Sender there is very invested in this research, and so he's asked to be a center. And due to limited funds for that trial, we can only open 3 sites right now due to the monitoring costs for each center, and that's where we are with that. So those are where our sites are open right now. If anyone wants more information on any of our clinical trials, Genevieve Bergendahl is our project director that oversees all the sites and kind of helps patients find their way to the closest center and where it's open. And that's her email. I'm always available as well, and there's my email and phone number if you have any questions. If you want any consent forms for any of the trials, you can just email Genevieve and she'll send you the consents if you want to read a little bit more about any of those. Online, we have our website at the nmtrc.org that talks a little bit more also about all our trials. And the support for the majority and all the infrastructure for our lab and the NMTRC is being provided by the NB Alliance right now, which Pat Lacey in the back of the room there has been critical in making that happen. And just in terms of acknowledgements, this is our clinical team. These are all the foundations that have been really helpful. None of this yet is government funded. We are trying very hard in getting grant funding, but really it's the families and parents that have made this whole program happen, and I can't thank you all enough for doing that. The Penelope and Sam Fund is our fund at the University of Vermont that funds all this, that gets donated there into the Penelope and Sam Fund. And I think that's it. Okay. So happy to take any questions.
Have some questions? Hopefully these microphones are working, because we want to make sure that we get the question in and everybody can benefit, and it can be taped as well. You know what? Use this.
Hi. Will you let the relapsed children be allowed to participate in the profiling?
Yes. It's only right now for relapsed patients. All our trials are focused primarily on relapsed neuroblastoma patients, because these are all experimental. Phase 1 and phase 2 is really only for relapsed or refractory patients at this point.
Okay, and the other question is, if Orlando, the hospital in Orlando, if it's doing this trial, and we are the sister doctor, like the Nemours in Orlando, I go to the Nemours in Jacksonville, would we be able to stay at Jacksonville, or would we have to transfer to Orlando?
You would have to go to Orlando for the biopsy, but the therapy could likely be done at your local hospital. Many of our trials right now, like our nifurtimox trial, the cyclophosphamide topotecan portion, we've been working with a lot of doctors around the country. So while you need to enroll at one of the sites, a lot of the therapy can be done at your home hospital. For instance, our DFMO trial, while patients need to be seen weekly, they only come back to us every 6 weeks for scans. Every week they're at home. And we definitely try and keep patients at home as much as you can. And most of the doctors have been wonderful to work with. Yeah.
Is DFMO methadone?
No, it's eflornithine is the other name for it.
Okay, thank you.
I think I know the answer, so this is probably going to be an unintelligent question, but what I'm wondering is, you said that the personalized medicine is done for relapse children. If your child isn't in that boat, hasn't relapsed, would it be at all smart or proactive to go ahead and get a personalized plan just in case, so that everything just moves a little bit faster, even though your 2-week turnaround period is extremely fast? But would it be faster to get it done ahead of time?
It's possible. We do have a biology study open, which I did. There are other things going on in our lab that I haven't had a chance to mention, but we have a biology study open. So anytime anyone here is ever getting a biopsy or has tumor tissue, we have a biology study that patients can enroll on, that we can do analysis of patients' tumors and do drug testing, expression profiling, and such. It's possible to do, but I would say that if the patient relapses, something's changed into the tumor to cause the relapse, and especially in the expression profiling is likely going to be significantly different, and would recommend redoing it at that point as well.
Okay, thank you.
Yeah.
Thank you.
Is that all? Okay. Oh, one more.
Can you tell us how varied the chemo regimens were for those 5 patients? I was just curious.
Yeah, we did have a couple of patients that were very similar. 2 that were very, very similar in terms of the final results. One, some of the drugs were similar, some of them were different, and then 2 that were completely different. So of the 3, there was some overlap.
Can you tell us how varied the chemo regimens were from standard induction?
Oh, very different. Yeah. So a lot of these targeted therapies, which we need to analyze further too. So there are definitely the targeted therapies we're seeing a lot more of that aren't necessarily used. So versus standard chemotherapy, kind of this. It's sorafenib, sunitinib, kind of that are targeting receptors specifically more than the DNA, which was interesting. Yeah. Yes?
Dr. Sholler, my question is, with the molecular guided therapy, can you get a biopsy on a bony lesion only that is not present in the bone marrow? I'm kind of beating the same drum here, Same problem with the ALK testing.
We have not tried that. And I think, some bony lesions, we do see a little bit of soft tissue extension on the outside as well. And I think, a good CT of the area to help us look and how much of the bone is involved is something we've talked about, but we haven't done on any patients yet.